Hi, I am Dr. Arsalan and today we are going to discuss the plasmids. Plasmid may be defined as plasmid is a small circular DNA molecule found in bacteria and some other microorganisms. Plasmids are basically small and circular type of the DNA. These are physically separate from the chromosomal DNA of the bacteria. So therefore plasmid is also called as extrachromosomal DNA because these are present outside the chromosome of the bacteria. As stated in the definition that plasmids are present in the bacteria and some other microorganisms including yeast and archae. This is a simple diagrammatic illustration of the plasmid in which a plasmid is shown as a circular DNA molecule having three regions: the antibiotic resistant gene, the promoter region, and the target gene or gene of interest, where the gene of interest is inserted into the plasmid. As far as history of the plasmid is concerned, it was discovered by Joshua Lederberg in 1952. He was an American molecular biologist. And he also won the Nobel Prize. Now we'll discuss some important features of the plasmid. That the size of the plasmid, it is highly variable depending upon the type of the cell. And the size of the plasmid may range from 1 to 400 kilobase pairs. Similarly, the number of plasmids per cell is again variable and depend upon the type of the cell. There may be one plasmid per cell, or there may be 1000 or more plasmids per cell of the organism. So the number is again variable, ranging from 1 to thousands. Now we'll discuss some important features pertaining to the plasmids. As you know that the plasmids are present in the bacteria. For example, this is a bacterial cell in which the chromosome is lying in a cytoplasm, where the region is called nucleoid region. This is the nucleoid region in which the chromosome of the bacteria lies. As you know that there is a single chromosome in the bacteria. Apart from this chromosome, there are some circular structures called plasmids. As these plasmids are physically separate from this chromosome, therefore these are also called as extrachromosomal structures or extrachromosomal DNA molecules. Plasmids are made up of double stranded of DNA molecule. So the DNA molecule contained in the plasmid is double stranded. The plasmids carry few genes, including the genes of the antibiotic resistance. The antibiotic resistant genes are present in the plasmids. Moreover, the plasmids can be transferred from one bacterium to the other bacterium by means of conjugation. For example, this is another bacterium in which plasmid is present. A conjugation tube is found between these two bacteria. This is also called as sex pili or conjugation tube. And the plasmids can be transmitted from one bacterium to other bacterium. It should also be kept in mind that these plasmids can replicate independently. So these are the self-replicating structures. Due to their self-replication procedure, these are also referred to as replicants. So the plasmids are also called as replicants. The plasmid has two origins of replication. Origin of replication present in the plasmid are two in number, persisting in their DNA molecule, and these DNA plasmids can also be artificially synthesized in the lab. And if these are synthesized artificially, artificial synthesis of the plasmids, such plasmids are called as constructs or vectors. So these are the synonyms of the plasmid: the constructs, the vectors, and the replicants. Plasmids are used in the field of genetic engineering, in which the gene of interest or new genes are inserted into the plasmids for replication and cloning procedure. These plasmids are also used in the field of biotechnology, as well as the biological production, in which the hormones, enzymes, as well as the vaccines are produced using these plasmid vectors. Now the advantages of the plasmids. As you know that the plasmids serve as tool to manipulate the gene of interest. The gene of interest or the desired DNA can easily be manipulated and replicated due to plasmids. Second advantage is that these serve as the factories to copy DNA or genes. As you know that the bacteria can replicate and divide at a very rapid rate, so therefore the plasmids also divide rapidly and can produce huge amount of the copies of DNA are gene of interest which are inserted into these plasmids. The plasmid, due to small size, can easily be manipulated or isolated from the organisms or bacteria. And subsequently, the genes can also be easily inserted into the plasmids. Due to circular configuration of the plasmids serve as a stable molecule as compared to linear DNA molecule. These can replicate independently, so called as replicons, and when the plasmid find a suitable host, these replicate independently in their host. These serve as the factory of DNA or gene of interest and produce huge quantity of the gene of interest or DNA which is inserted into these plasmids. And finally, the examples of the plasmids. The RNA plasmid, TI plasmid. TI means tumor inducing plasmid. PBR322 is a notorious and common plasmid used in the genetic engineering and biotechnology. PBR means Plasmid Bolivar and Rodriguez. It's named after the scientists, the Bolivar and Rodriguez. Then the plasmid PUC 18 and PUC 19. PUC means Plasmid University of California, because most of the plasmid PUC were used in the University of California, so these were named after this institute. These were some information regarding the plasmid. Hope this lecture will bring fruitful results to you. Keep watching my lectures. Thank you.